Great. Thank you for the introduction, and thanks so much for the opportunity to speak today. I'm so sorry I couldn't be there in person. I'm definitely sorry to miss the photo sun. But yeah, as mentioned, I'm a machine learning researcher here at Microsoft Research in Cambridge, UK, and I'll be talking today about our work on the Antigen Map project on decoding the adaptive immune system. So for completeness, I'm going to start with a quick background on the basic underlying biology, so please bear with me. So T cells are the core component, of course, of the adaptive immune system, responsible for identifying and fighting disease. Let's consider how our body responds, for example, to a viral infection. A virus will have these protein regions, disease antigens that are responsible for eliciting an immune response. And there are dedicated antigen-presenting cells in the body that consume these proteins, break them down, and present them on the cell surface. And then T cells come in. Each T cell has distinct T cell receptors, or TCRs, that will bind very specifically to the presented antigen. And a binding event triggers a signaling cascade that causes that T cell to make copies of itself. So it triggers clonal expansion. The DNA sequence, the DNA sequence that encodes TCRs is distinct for each T cell, and there's a massive diversity in TCRs that can be generated, more than enough to target all known antigens. And we can measure the expansion of a given TCR by counting how many times we read its DNA sequence in a blood sample. It's all very basic. So what we're hoping is that the state of our immune system can, in theory, provide an account of all past and present disease exposures, if we can say which TCRs are specific to which disease antigens. And this is basically the motivation behind the Antigen Map project, the development of T-cell-based diagnostics. So the overall pipeline looks something like this. We take blood samples from various individuals with known exposure status to the disease we're interested in. From these blood samples, T-cell receptors are sequenced and counted. This data set of repertoires and disease labels is passed through a machine learning model, and that allows us to classify individuals as having or not having disease, hopefully empowering care providers downstream. And we think that we're hoping that this is possible not just for infectious disease, but conditions such as autoimmune disease or cancer. Taking a step back, let's consider some of the ways we're currently diagnosing these diseases. This is, of course, an oversimplification, but shown here are the primary methods for confirming disease in various cases. These approaches remain limited in a number of ways. Just to name a few, they're often quite invasive and expensive. Things like endoscopy or biopsy require multiple hospital visits and regular follow-ups. They can be unreliable. Current tests for Lyme, for example, have a false negative rate of 20 to 30%. They're often sensitive to the timing of the test. We know PCR tests can be very accurate, but cannot detect past infection. Whereas in many conditions that have long-term health outcomes like Lyme or COVID, analyzing this lingering immune response could be of interest. And diagnosis often comes too late for conditions like multiple sclerosis or cancer and many others. Diagnosis based instead on TCR response would not only be relatively non-invasive and inexpensive, but can also potentially be highly sensitive to any small amounts of disease, as well as highly specific. The underlying mechanism they rely on means that these small specific signals of disease are systemic and naturally amplified through the clonal expansion process. And finally, that signal is persistent, with the immune system retaining long-term memory of these disease exposures. The fundamental approach we take here to building such a diagnostic is pretty simple. Taking COVID-19 as an example, we first curate a data set of COVID-positive TCR repertoires and healthy controls. For each of the millions of TCRs in these repertoires, we run a statistical significance test based on the presence of that TCR in cases versus controls, namely Fisher's exact test. We select TCRs of interest based on some threshold p-value, and then we aggregate the counts of these selected TCRs, or enhanced sequences, and we fit a logistic regression model to separate cases and controls, taking this enhanced count as input. This can basically be viewed as a kind of multiple instance learning problem. We have a bag of instances, or TCRs, and that bag is labeled positive if some number of instances in the label are positive. In March 2021, Adaptive released an FDA-approved test for COVID-19, which was an exciting milestone for TCR-based diagnostics. And so this all seems quite promising, but what then are the challenges? The complexities of immunomics data begin with, of course, what's inside an individual blood sample from which Adaptive sequences and counts these TCRs. This repertoire of sequences will contain between 100,000 to 1 million unique TCRs, and these TCRs are going to be a mixture of active, so what we're currently fighting, memory, so the immune system's history, including vaccinations, and naive T cells, T cells that are just patrolling your body and have yet to find their cognate antigen. And so isolating the active TCRs that we care about for our diagnosis, for example, can be pretty difficult. The disease-specific TCRs we see in any given individual are also partly determined by things like their age, genotype, treatment, such as chemotherapy or immunosuppressants, and so on. So the observed response to disease can be highly heterogeneous across different samples. And though this may vary a bit with disease, we're expecting only of the order of about 100 to 1,000 TCRs in a repertoire to be associated with the disease of interest. So this is going to be an extreme small n large p problem that requires careful treatment to pull out robust signal. Confounding can also, of course, obviously come from external factors. We currently have access to sample repertoires for over 100,000 individuals across more than 100 different sequencing collections or cohorts with 20 or so different diseases. This is probably an underestimate at the moment. Each repertoire brings with it its own set of potential biases that we need to control for in order to identify disease-specific signal. For instance, Lyme disease is endemic to certain regions of the US, so we would need to carefully match controls to avoid false positives in these endemic regions. Certain diseases may also come with associated genetic risk factors. For example, we know there's a risk allele that is a prerequisite for celiac, which shapes the immune repertoire significantly, but does not determine the presence of the disease. And age could also be an important confounder, as with T1D, which is skewed towards children, and certain cancers, which could be skewed towards older people. And finally, we see systematic batch effects from various labs that source our data and differences in sequencing protocol versions, which may crop up incorrectly as signal, especially since our cases and controls tend to come from different labs. The second limitation I want to mention here is the loss of private signal. In our current approach, the millions of TCRs per repertoire are treated independently, agnostic of the actual protein sequence that makes up the TCR. TCR selection for downstream classification therefore relies on an exact match of a subset of sequences observed to dominate the response to the same antigen in multiple individuals. But in practice, this often only makes up about 10% of the disease response in any given individual. That means we're throwing away the other 90% of what we're calling here private signal, limited to a small number of individuals, since with our current data, we typically need at least about 7 people to share a TCR in order to pick it up from FET. If we assume that these private TCRs are in some way structurally similar to public TCRs, however, public TCRs that are responding to the same disease antigen, this motivates approaches that explicitly model sequence structure to pull out these rarer TCRs. The primary region of the TCR that we think is responsible for binding, the CDR3 region, is highly structured, a short sequence of between 10 to 30 amino acids in length. And this is pretty unique to what was the majority of existing work on protein modeling for many years. Okay, so returning to the problem of handling confounding in the data, the question becomes, how do we reliably identify disease-specific TCRs in the presence of confounding? There are a number of different approaches we could take to this. For example, we could just try to refine the statistical tests we're using for sequence discovery. We could apply algorithms for group robust optimization to balance performance across different subgroups. Or alternatively, we could try to model the data generating process explicitly to disentangle these confounders. And this approach has been the focus of a lot of our recent work. Specifically, we developed what we call an adaptive immune repertoire-invariant variational autoencoder, or AIRRIVA. What this is essentially a partially supervised variational autoencoder, which aims to learn a factorized low-dimensional representation of the TCR counts. So the TCR counts are the input X, and we have labels Y for the disease of interest, as well as any other relevant factors of variation. So this low-dimensional representation basically comprises these predictive latents, ZY, that are specific to each of the labels provided, and residual latents that are used to reconstruct the rest of the TCR repertoire. As we will show, what we're hoping is that AIRRIVA presents a unified probabilistic framework for firstly predicting repertoire labels given TCR counts, generating TCR counts conditioned on specific labels, and finally estimated the causal effect of intervening on a given label on the observed TCR counts. So let's turn back to COVID-19 in this example. The viral envelope of SARS-CoV-2 is made up of 3 proteins. So the membrane protein, the envelope protein, and the spike protein. The spike protein is what we believe to be responsible for binding to host cells and initiating viral infection. And so that's been the target of vaccination development. As mentioned, Adaptive released this TCR-based COVID diagnostic in early 2021, but what we found was that when vaccines became widely available, there were an increased number of false positives from vaccinated individuals. This basically motivated the need to distinguish the response to the spike protein, seen in both vaccinated and COVID-positive individuals, from TCR responses to non-spike proteins, which only occurred in infected samples. So based on labels for ground truth disease and vaccination status, we can train AIRRIVA on derived spike, non-spike labels. Here, I'm trying to visualize the learned latent representation that we get from AIRRIVA, specifically the latents that are corresponding to the spike on the y-axis and non-spike on the x-axis, respectively, which shows that the model has learned to disentangle these 2 components of TCR response, despite their correlation in the data. So that is, for example, you can see that controls here are at the top top right of this space, and as you navigate along Z spike, we get to along the y-axis to Z spike, we see vaccinated individuals, where we have activation of just the spike response. As we move across the x-axis along Z non-spike, we now see natural infection, samples of natural infection, so both spike and non-spike. The top right here corresponds to non-spike only samples, which obviously we don't observe in practice. Given such a disentangled representation, we can use the trained model to answer counterfactual questions about the data. So the procedure basically looks something like this. We pass the repertoire through our encoder to get the low-dimensional latent representation. We can then intervene on on a label of interest and sample a new value for that predictive latent accordingly. And then we pass our intervened latent through the AIRRIVA decoder to reconstruct a new set of TCR counts corresponding to these intervened labels. So this basically, what this lets us do is answer questions like, what if we switch disease on and off? What if you change the presence of a risk factor? How is this going to change the counts that we observe? Importantly, we can also use these counterfactual repertoires to evaluate treatment effects. So that is the effect of a label on each TCR. To evaluate the individual treatment effect of the label in a given repertoire on some TCRI, we can simply take the difference between the factual and the counterfactual counts for that TCR with the label on and with the label off. And then if we take the mean across all repertoires, this allows us to estimate the average treatment effect of the label. And if we sort by average treatment effect, we can try and pinpoint true disease-associated TCRs. Again, we can illustrate this in the context of the spike, non-spike task in COVID-19. So in addition to labeled repertoires, within the antigen map project, we have access to a limited amount of biological assay data, so MIRA, as a source of ground truth TCR antigen annotations. This provides us a way to validate learned TCR associations from ATE estimates with AIRRIVA. So naively, in the absence of a model, we could obtain an estimate of treatment effect by simply taking the difference in the mean counts for each TCR in all observed cases versus controls. With respect to each label, so spike and non-spike in this case. The figure on the very left shows ATEs that are calculated empirically in this way. We can see from here that it's incredibly difficult to distinguish MIRA hits for spike-specific TCRs, so the red stars, from those for non-spike, so the black squares. Alternatively, if we use domain knowledge selectively to choose informative or better match cases and controls with respect to the spike and non-spike labels, for example, for spike, we just use vaccinated individuals versus controls, and for non-spike, we just use infected natural infection versus vaccination. To evaluate those non-spike ATEs, we can see much better separation. This can be thought of as a kind of oracle, that we find that matching case controls in this way gives rise to different biases. It has limits to how far we can subgroup like this, and we can see that, for example, in this case, we end up with a slight negative association between the spike MIRA hits with respect to non-spike. Instead of using AIRRIVA to generate counterfactual repertoires, Instead, if we use AIRRIVA to generate counterfactual repertoires with respect to the spike and non-spike labels, using all of the same data and labels as the naive approach, we can effectively separate spike and non-spike with almost entirely decorrelated ATAs. So the spike response all along and non-spike response all along their respective axes. Okay, so changing tack slightly, I wanted to talk about how such a model could also be used to address the second challenge I posed at the start of the talk, so that of identifying private signal from rare TCRs by modeling sequence structure. Fundamentally, what we want to do is learn a low-dimensional TCR embedding that holds some predictive power for TCR antigen specificity while correcting for confounders, such as difference in generation probability of the sequence in the underlying biological process, to name one possible source of bias. To do so, we posit a variation of AIRRIVA that takes as input the sequence of amino acids in an individual T cell receptor, as well as per TCR level labels where we have them, that we expect these to be pretty noisy. Here's a toy setting to illustrate this model. Okay, as we know, TCRs are generated from a process called VDJ recombination. The CDR3 region is thought to be is thought to primarily drive specificity is determined by the V, D, and J genes. V and J are almost perfectly recoverable from the sequencing process, so we kind of have these sequence level labels, and we can use them as a proof of concept for learning a sequence embedding with some supervision. Shown here is the latent space with categorical J gene label on the x-axis and length on the y-axis. CDR3 length on the y-axis. Again, so I'm coloring points here by J gene label, and we can see that we've managed to learn an axis-aligned representation where the sample J gene for that particular sequence changes as we travel along the J latent axis. We also find that we're able to generate sequences conditioned on the J gene quite faithfully. We know from the biology that J determines the end of the sequence, and we see that this is the variable part when conditioned on different J gene labels. We also see that generated sequences correspond reasonably well to the empirical distribution for this J gene, although we do see a little less variability. I think this has come up in previous talks. And given these results, what the hope is that the next step is basically to determine what TCRs we should train on, possibly how we tailor the losses to ensure diversity in the sequences generated, and which labels to use in order to learn informative representations that tell us something about TCR antigen specificity. So this brings us to the question, given some learned sequence embedding, how do we go about evaluating its utility? Thus far, our evaluation approach has been founded on our end goal of developing improved models for disease classification. As a reminder, our basic framework for building diagnostic models is based on selecting a set of TCRs based on FET, aggregating the counts for these TCRs in the repertoire, and learning a classifier with aggregated counts as the input. We can generalize this multiple instance learning setup as illustrated here. So first, we embed TCRs into some latent space. Then we aggregate these latent features, weight them by their counts, and then apply a discriminator, like logistic regression in the simplest case, on top of this. Rather than learning this embedding from scratch, we could first train a sequence encoder using the generative seq-diva model, taking as input all available sequences and relevant sequence level labels. And then we could use this to initialize the embedder component in multiple instance learning and fine-tune to the task at hand. This mode of generative pre-training followed by discriminative fine-tuning has been proven effective in other settings, and we're hoping to translate this here in a domain-specific way. There remain several challenges in developing such a model, of course. What are the assumptions, for example, that we need to make about the underlying causal graph in order to trust the counterfactuals from our model? In what way might we need to either constrain or increase the flexibility of our models to make this possible? And how do we handle the high degree of label uncertainty in the data, especially for seq-diva? And finally, what is the right way to provide labels? And finally, how can we scale these models to leverage the order of 10 billion TCRs, 100,000 samples, and hundreds of labels available to us towards a kind of foundation model for TCRs? Before I finish, I wanted to just mention some ongoing work that's taking a fairly different approach to generative modeling of the TCR peptide MHC complex based on diffusion models. So there was already a short introduction to diffusion in the keynote earlier, but just to give a quick review. So diffusion models are a class of generative models that generate data by iteratively denoising a sample of pure noise. So they've gained a lot of attention for their ability to produce high-quality, realistic data, particularly in image generation, but their applications could potentially extend, of course, to biological sequence generation. They've been used certainly for 3D protein structure prediction, but their use in sequence modeling is to date sort of relatively limited. There are a couple of key concepts to consider when we're training diffusion models. So we have first the forward diffusion process, which we specify. So this process basically involves gradually adding noise to the data over a series of steps, transforming the data into a distribution of pure noise. And each step basically incrementally corrupts the data, creating a smooth transition from the original data to noise. And how we do that corruption is up to us. Then we have the reverse diffusion process. So the generative process basically works in reverse, starting with a sample of pure noise and iteratively denoising it to recover the original data. This denoising function is basically the function that we want to learn. In the case of protein sequences, there are different ways you could design this noise function. For example, masked corruption, where positions in the sequence are iteratively masked at each time step, or with discrete corruption, where amino acids are instead flipped with some probability. So total noise is just a sequence of random amino acids. So diffusion models could offer several potential advantages over the sort of VAE frameworks that I've described up to now. They're capable of generating very high quality and diverse samples. For TCR or peptide or HLA sequences, this means producing sequences that are not only syntactically correct, but also functionally diverse, covering a wide range of potential interactions. While VAs can generate sequences, they often struggle with maintaining diversity and functional correctness. Diffusion models can better capture the wide range of variability. Variability in TCR and peptide sequences. The training process of diffusion models tends to be more stable. The structured denoising process helps in learning a robust representation of the sequences, whereas VAEs, particularly the partially supervised VAE I described here, can be tricky in terms of balancing the various components of the loss function, particularly when we are aiming to do both reconstruction and prediction. Balancing these various losses can be difficult to achieve, and this can lead to potential issues in the generated sequence quality. And diffusion models are well suited for handling complex dependencies and offer a lot of flexibility in doing conditional sequence generation. And this flexibility is beneficial for tailoring the generation process to specific biological requirements for various downstream applications. What we're proposing here is a multimodal diffusion model in which each component of the TCR peptide MHC complex is modeled by a discrete diffusion process. So we start with a fully masked sequence, then generate amino acids either sequentially or at random positions at each time step. Each mode, so like the HLA, the peptide, the TCR beta and alpha chains, can be pre-trained independently. The important part here, or the tricky part, is the binding prediction, how to tie these together. Let's restrict ourselves, for example, to considering just peptide HLA binding. For learning peptide HLA binding pairs, we want the generation of the peptide at the next time step, for example, to consider not just the peptide at the previous time step, but also the HLA and vice versa. This could potentially be achieved through some kind of gated attention mechanism. For non-binding pairs, on the other hand, we would want the peptide and HLA diffusion processes to be independent. To learn this gated attention mechanism, we can learn another popular approach in recent literature based on contrastive learning to train a clip-style projection. So if we have an embedder, like a transform-based model for the peptide, the HLA, and so on, we can learn projections on top of these embeddings such that to map them both into a single projection space, such that a peptide embedding and a HLA embedding, binding pairs are basically mapped close to each other in this projection space, and non-binding pairs are far from each other. Ultimately, the hope is to have a model that firstly is able to consider the entire TCR peptide MHC complex, is able to learn from both the any unimodal data that we have, since we have a lot of it, as well as the limited amount of combinations of multimodal data or weakly labeled data. The model should be able to predict pairwise binding, as well as allow sequence generation with arbitrary conditioning. And given a peptide, for example, if we're given a peptide in a HLA, we want to be able to suggest a binding TCR beta chain, or given the TCR in the HLA, we may want to generate candidate peptides. And such a framework should potentially be able to achieve these, but there are a lot of open questions, and I'd be happy to chat more offline if anyone's interested. I'm going to stop there. Thank you all for listening, and I just want to thank the amazing collaborators in my immediate team, as well as in the wider Anshin Map project. I'm happy to take questions now.
Thanks, Niranjani. We're a little bit over time, so if you could very quickly just answer a couple of questions for us from the Slido. Could you please clarify what the inputs and latent outputs of your autoencoder are? A short example would be great. Thanks.
Sure. So let's focusing on the repertoire level autoencoder that I spent most of my time talking about. The inputs are basically a set of counts. One limitation of AIRRIVA is that we still have to choose some subset of TCRs that we are going to input in terms of their counts. So if we have a set of 100 counts based on the top 100 from FET, we put those in, and ideally, we would like to be able to put 1,000, 10,000 TCRs, the counts for those TCRs based on their index in the repertoire as inputs, and still be able to filter to those that are allow the model to learn which ones are most important for certain tasks. But yeah, it's still quite sensitive in terms of how many TCRs they can take as input. The labels are the outputs are the disease labels. So for example, in the case of COVID, it's disease and vaccination status, for example. And it's a generative model, so we could potentially regenerate those counts. So if we have 100 TCRs that we pre-selected, we don't have any knowledge of their sequence structure. We just know that they're already enhanced in cases versus controls. The idea is, given that we want to generate a vaccinated sample, can we generate what the expected counts for those 100 TCRs are going to be, given that some of those are spike TCRs, some of those are non-spike TCRs, some of those are neither.
Great, thanks. And finally, could you touch on how the model learns the invariance and how it's defined? Is it invariance over haplotype, antigen exposure, sex?
Okay. The goal of the model is basically that, given that since we have these latents that are intended to be able to predict certain disease labels, the information associated. So so for example, if one of our labels is HLA, or if we have labels for HLA or for cohort, lab, categorical label for lab, for example, if we're able to predict those well, all the information in the sample that is associated with that label should be captured by that particular latent, and should not leak into other latents, for example, the disease latent that we're actually interested in. Obviously, yeah, so even if there is a correlation between these two labels, we should be able to use Z disease to just predict disease and not be influenced by other labels, because reconstruction of those other properties of the sample is already possible with Z haplotype, for example.
Fantastic. Once more, let's thank Niranjani.